Okay, there's a little inconsistency between my class periods between 2, 4, 5, and 6. So what I thought I would do is I would do a little bit with some organic chemistry just to make sure everybody's on the same page. And from there, we'll talk a little bit about some of the functions of these macroorganic molecules. So I think to start off with, we better just review very quickly the 4 main categories of macroorganic molecules. The first category that we'll talk about are carbohydrates. And carbohydrates are actually molecules that act as a good energy source. They also act as energy storage. This in class. Monosaccharides that are important in the human body include the hexose sugars. Hexose sugars include sugars that have 6 carbons, and they have the formula C6H12O6. They include glucose, fructose, and galactose. Now, these 3 are really pretty much just used as a source of energy. That's their main job. Now, hexose sugars aren't the only sugars that are found in the human body. Another important group of sugars are pentose sugars, and they have the formula C5H10. Whoops, that's supposed to be an H. C5H10O5. And the 2 that are important are ribose and deoxyribose. You may have heard both of those. Deoxyribose is the D for DNA, and ribose is the R for RNA. So that's where those particular sugars are important, or where they come into play. There are other sugars like triose sugars that are 3-carbon sugars, and there are sugars that are hexose sugars, pentose sugars, and they even go higher than that, up to 7, 8, 9. But in the human body, the 6-carbon sugars, hexose, and the pentose sugars, the 5-carbon sugars, those are the most common ones that we'll be talking about. So again, monosaccharides mostly for energy. Now, using a reaction called a dehydration synthesis, we can actually take 2 monosaccharides and attach them, and what you end up when you do that is a disaccharide. And there are 3 disaccharides that are important in human physiology and in human nutrition. One of them is called sucrose, commonly called table sugar. You get sucrose by taking a glucose and dehydration, synthetically reacting it together with a fructose. Another important sugar is lactose. Lactose is also known as milk sugar. This is also a glucose, but in this case, instead of a fructose, this is dehydration synthesized with a galactose. And then finally, maltose, which is a glucose attached to another glucose. Again, these are mostly energy molecules. They do need to be digested before they're useful to the body, but they're easily digested and quickly digested, so that's why they are also considered kind of an energy source. As we continue down, we end up with polysaccharides. And so if we kind of back this up a little bit, this is going to follow a trend that's going to be kind of happening with all of these different kinds of macroorganic molecules. The monomer, the building block, are the monosaccharides, and the polymers are the polysaccharides. The disaccharides are kind of a bridge between them, although that's not entirely true because polysaccharides are almost entirely made of glucose, where lactose and sucrose have those oddball fructose and galactose involved. As far as polysaccharides are concerned, we've already mentioned this in most classes, but plant starch is a significant energy storage, and animals have their own form of starch called glycogen. And I will remind you that plant starch is also called amylose. That's a term that's not used too often, although our textbook does mention it. Glycogen is animal starch, and then finally cellulose, which we mentioned in class as a form of fiber, which is important for the digestive system's functioning, although it doesn't have any nutritional value for us. We can't digest cellulose, and that's why it passes through as fiber. So glycogen, energy storage, plant starch, it is an energy storage molecule for plants, but when humans eat it, it's a ready source of glucose. So all of these are basically glucose molecules strung together one after another, making large chains, and that's why they're called polymers, and then again polysaccharides. Okay, so that takes care of carbohydrates. Let's see if we can then continue on with something we'll call these lipids. We'll do that next. Lipids are somewhat unique because these guys really aren't true polymers. So they don't have that monomer slash polymer relationship. They don't have true building blocks, but they do have some commonalities. Most of them, if not the entire molecule, most of the molecule is hydrophobic, which means that it's water-fearing. Water-fearing, alrighty? So we've got three main groups that you need to know something about as far as these polymers are concerned, or these, not polymers, these lipids are concerned. And one of them would be fats. Fats are also known as triglycerides. Triglycerides are made up of a glycerol. This is a 3-carbon alcohol, and attached to the 3-carbon alcohol through special covalent bonds called ester bonds, you end up attaching 3 fatty acids. These molecules make up what are known as fats, or traditional fats. These are good for energy. They're good for heat insulation. They're good for cushioning. So fats are a very important part of the body. In fact, various parts of your body has to have fat, otherwise it doesn't function properly. Kidneys being one of them. It's the adipose tissue, the fat tissue, that actually holds your kidneys in place. So you definitely do need these things. So fats would be one type of lipid. Another important lipid would be phospholipids. Phospholipids are made much the same way. You do need a glycerol. And to that glycerol, we attach 2 fatty acids. So you have a glycerol, 2 fatty acids, and then instead of a 3rd fatty acid, which would make it a triglyceride or a fat, we add a phosphate group. Now the textbook is going to draw these things in cartoon nature like this. That would be the phosphate head. And then there's going to be fatty acid tails dangling down. The glycerol molecule will be a little backbone in here, but they hardly ever draw that, so you never see the glycerol in there, but there is a glycerol in there as well. The unique thing about a phospholipid is that this orientation, this phospholipid, is actually an amphipathic molecule, which means it actually has a dual nature. You hear the word amphipathic and you hear the word amphibian, and they have a similar nature. It means dual life or dual nature here. This top part, the phosphate, is actually hydro, and not phobic, but philic. It actually is water-loving, where the fatty acid tails are hydrophobic. So that's unique nature of phospholipids. Phospholipids are somewhat unique in that dual nature. Most of the molecule is hydrophobic, so it kind of still fits in that lipid category, but it is unique. So that's our second type of lipid. Our third type of lipid, basically the last one that we're going to focus on here, although there are other types of lipids, that would be steroids. Steroids are large molecules, as all of these are, but steroids are ringed structures. So steroids always start off with carbon rings, and they're usually in a hexagon kind of arrangement, something like this. Now, I'm not going to draw the carbons in here. They're a little more complicated than this, but they usually tend to go up something like this, and then they usually have a 5-carbon one on the end. So you have that 4-carbon ring. It's usually a 4-carbon rings. If you think of that kind of looking like the Olympic rings, you can kind of remember that Olympic rings and steroids don't go together. Maybe that'll help you identify these things. But what I will tell you is that these are much more complicated than I'm drawing them. There are carbons in here like this, and there are double bonds in various areas. But essentially, all steroids have this general shape or this general style. The steroid that I've drawn right here is a steroid known as cholesterol. And cholesterol in the human body acts as the basis for all other steroids. So cholesterol is a very important steroid. And again, steroids then being our third and final type of lipid. Now, there are other lipids. There are prostaglandins that are important, but we'll talk about those on a case-by-case and specific situation. So that's the second of the 4 macroorganic molecules. So we had carbohydrates first, and that would be lipids. Okay, let's clear this page. Get rid of that. Okay, that brings us back then to a fresh page. So we've got carbohydrates done, we've got lipids done, And now we're back down here to, let's go with proteins next. So proteins have the most diverse functions. I guess I didn't give you some functions for phospholipids. I thought it was kind of self-explanatory, but maybe it's not. Phospholipids would be important in cell membrane function. We're going to look at a picture in our study guide in just a second on that, so I'll leave that one alone. And then the steroids have functions as hormones, among other things. Cholesterol is important in cell membranes as well, and we'll get to that in a second. Proteins have a ton of functions. So proteins are involved in all sorts of things. Now, this is not the only list of protein functions, but it's a list that I'll just kind of start here for you. Proteins can act as organic catalysts and act as enzymes. They're going to speed up chemical reactions. Enzymes can be involved in defense. These would be like antibodies. Proteins can be involved in transport. We move O2 with hemoglobin. That's not an example of transport. We also use proteins in membranes to transport ions and other things across cell membranes. So there's another example of a protein for transport. Movement, muscle movement, large muscle movements. Those are all caused by proteins in the muscles. We also move things inside cells. So chromosomes, when they're moving during mitosis or meiosis, are moving on proteins. So proteins are involved in that movement as well. We also have proteins doing things like energy. Proteins can be used as an energy source, although that is not something that they're common for. Carbohydrates are a much better energy molecule, but you can use proteins as an energy source, just not a very good one. Now, proteins come in two major types. We have structural proteins. Structural proteins all have a three-dimensional shape, so we say that they're globular. And, whoops, I said that backwards. I'm going to back this up. I'm sorry, not three-dimensional, not structural. I'm sorry, structural are fibrous. And the functional are three-dimensional or globular. So the hemoglobin molecule, the antibody molecule, the enzyme, all of those would be functional. So those would all be underneath here. Enzymes, antibodies, hemoglobin, all of those would fit there. As far as the structural ones, and I didn't list that as a function over here, but I could have. The structural ones involve things like collagen and keratin. These are structural proteins. These are fibrous or filamentous, long and thread-like instead of globular. Now, proteins are made up of building blocks, or again, monomers. We're back to our monomer situation, and the monomer is known as an amino acid. Amino acids all have the same beginning formula. You have a central carbon, and off of that central carbon we have an acid group. This is a functional group called a carboxyl group. And this little hydrogen is or will fall off in solution, releasing a hydrogen ion, so that makes it an acid. Now I'm going to get rid of that so it's not confusing. I'll leave it as a carboxyl group. And then we have on the other side an amino group. An amino group, or an amine group, is a nitrogen-containing group, so there's the nitrogen there. Now in the central carbon you have a hydrogen, and that's always there, and then there is an R. This is a variable. This can change between 1 and 20 variations. The simplest variation would simply just be an H right there, and that would be an amino acid known as glycine. You can change that into a variety of different structures. Some of them are very complicated and complex, but I'm just going to leave it as an R group, so we'll have that down. So that is an amino acid. Now, what we can do with these amino acids is we can join them together, and I'll show you on the next page how that happens. But when you join them together, if it's just two, it becomes a dipeptide, then a tripeptide, but eventually, when you get a number of them strung together, they will end up being a polypeptide and eventually a full-fledged protein. So proteins fit the model for macroorganic molecules because they're quite large. They also fit the polymer and monomer model because they have monomers named amino acids, and then they have polymers known as proteins. Or if you're really into science, polypeptides. We usually call them that when they're smaller before they get too large. Now, the book does talk about different levels of protein structure. Proteins can be structured into various levels. The first level would be primary, then there's a secondary, then there's a tertiary and a quaternary level of protein structure. That's a little beyond the level of what we need to understand for this class, but if that's something you're interested, don't hesitate to come in and I can go over that with you if that's something you would like to know more about. So that's our third type of macroorganic molecule. Our final one is going to be nucleic acids. We're not going to spend much time with nucleic acids until later in the year when we do a little bit of genetics at the very end. Chapter 29 will have a little bit of genetics involved. But nucleic acids are polymers, so that is a polymer. The example would be DNA and RNA. And it's made up of monomers called nucleotides. And these nucleotides have a very distinctive structure. They're very easy to identify. They always have a 5-carbon. We talked about that pentose sugar. There's 1, 2, 3, 4, and then there's a 5th carbon coming up like that. So this is a pentose sugar. If there is a hydroxyl group here, it's called ribose. If you take off that little oxygen right there, it's called deoxyribose. So I'm going to leave it as deoxyribose just because DNA is something that most people have heard more about, so we'll leave it as DNA. There's a little OH on this side. There would be other OHs and H's there, but I don't want to complicate the drawing anymore. So this is a 5-carbon sugar called a pentose sugar, and again, specifically, this is deoxyribose because of that missing oxygen right there. Attached to that, there is a ringed structure, and this ring structure can be a simple single ring, like I've drawn there, or it can have a double ring. Single ring, double ring, that is a difference in between one nitrogenous base and another, and the nitrogenous bases are adenine, guanine, cytosine, and thymine. Now if we're talking RNA, we'd throw uracil in the mix, so we do have some differences there. And then off the top here, you have yourself a phosphate group. Now, this is a nitrogenous base, which means that in that double ring structure there are nitrogens in there. So I'm going to throw a couple nitrogens. These are not in their real location. I'm just throwing them in there for an explanation. So we do have some nitrogen there. We do have some phosphorus there. This is a single nucleotide. This is a large molecule all by itself. Now, you can imagine how large DNA must be, because to make the polymer DNA, you need to string a whole bunch of these together. And as you remember from sophomore biology, these things are strung together in 2 strands where you end up with a double helix where they're held together with hydrogen bonds in the middle. So just to kind of recap then, we can recap this whole thing by saying the macroorganic molecules, there is the monomer and polymer relationship. If we're talking proteins, the monomer are amino acids and the polymer would be proteins. If we're talking nucleic acids, the monomer would be a nucleotide and the polymer would be the large molecule like DNA or RNA. If we're talking carbohydrates, the monomer are monosaccharides like the hexose sugars, glucose, fructose, or galactose, or the pentose sugars like ribose or deoxyribose, and then the polymers would be polysaccharides like starch, glycogen, or cellulose. And then our final one that doesn't fit the monomer-polymer relationship, so we got to kind of draw the line there, and that would be lipids. What they have in common is that they're all hydrophobic, and so when you have your fats, you have your phospholipids, and you have your steroids, you have what you basically have are hydrophobic molecules. So that will conclude the discussion on macroorganic molecules. We'll have another discussion here on how some of these things function, but that should get you a good start if you're confused.